So today our next video is the chemiosmosis and oxidative phosphorylation, which means that the complex we are having that will get oxidized. No, we are having the two things, we are having NADH and FADH, they will get oxidized. Okay, and the coenzyme we are having that will be reduced. I'll be making diagram, you can easily understand from that. And that through which the you can say that the we are having a particle which is called F0 and F1 particle, through which the ATP got synthesized. We are calling that as oxidative phosphorylation. In the end of the video you will easily understand what I'm talking about. Okay, so this is the inner membrane. This is what? This is the inner membrane. Inner membrane of the mitochondria. Okay, this is the intermembrane of the mitochondria. As you guys all know, intermembrane of the mitochondria. Okay, if we talk about this, so this is the mitochondria matrix, which is the fluid. Mitochondria matrix. Okay, which is the fluid. Okay, if we talk about that, we are having the mitochondria fluid, which are having the inner membrane mitochondria. This is the intermembrane mitochondria. Okay, so we are having many particles. As you guys all know, we are having the, for example, we are having the you can say the electron carriers. Okay, we are having that is complex 1, as you guys all know. In the previous video I have discussed you guys about the, this is the complex 2, this is the complex 3, and this is the cytochrome C, and here we are having that is complex 4. Okay, and in matrix we are having the hydrogen proton hydrogen proton. So complex 4 will be the final acceptor. That is these complexes are what? These are the protein. This will be the final acceptor, and you can say that the final acceptor will be the protein. If we talk about the molecule acceptor, that is the oxygen with through which the hydrogen will be added and making a H2O, that we have discussed in the previous video. Okay, so the concentration is increased in the mitochondria matrix. Okay, so the you can say that, so these hydrogen proton will be pumped into the intermembrane space, like it will be moved into the intermembrane space. Okay, so we are having the three sites where the you can say that the hydrogen will pumped first in the one, and second in the third one, and fourth is the fourth one. Okay, as you guys all know, now the hydrogen is moved into the intermembrane, so the intermembrane is having the more concentration, and if we talk about the matrix, so the concentration is less in the you can say that the of the hydrogen proton. Okay, so when the electron is basically, you can say that the electron passage is occur. Okay, so the energy is basically released, so the hydrogen ion pumped from mitochondrial membrane to the mitochondrial matrix to the intermembrane space. So in this we are having the three you can say that complex where the proton will get pumped. We are having three sites. First one is called the NADH dehydrogenase complex 1. Second we are having that is the cytochrome reductase complex 3. Fourth one we are having the cytochrome oxidase complex, which is fourth one. Okay, so this inner membrane is having one more particle that we are calling as a F0 or F1 particle. Okay, so this particle, so this FO will act as a channel, and F1 will be containing a ATP synthase enzyme. Likewise, let me make the structure through which you will get easily that what the thing I'm talking about. Okay, so this is the F0 particle. Okay, and this is the F1 particle. Okay, F1 particle is having the enzyme that is ATP synthase, and F0 is the particle. Okay, F0 is the particle which will be act as a channel. Like here, this is the channel. This will act as a channel. Okay, so what is, now you will easily understand what is oxidative phosphorylation. Oxidative phosphorylation is basically the formation of the ATP when the NADH, here the NADH and the FADH were oxidized, and you can say that they oxidize and this complex was getting reduced. In the previous video we have discussed, so they will release the energy, that we are calling as the oxidative phosphorylation. This is the fifth particle we are having, that we are calling that. Okay, so here you can see that we are having the ATP synthase through which the ATP will be formed. So this ATP synthase where the ATP will be formed, that we are calling as the chemiosmosis or the oxidative phosphorylation. So as you guys all know, we were having the 8 NADH, and you can say that 2 FADH, and 2 NADH, the final product we were having. Okay, so ATP, so we were have this 8 we were having from the, you can say that the Krebs cycle and the length cycle, we were having the 8 ATP, and FAD were having the 4 FADF was there and 2 NADH. As you guys all know, 1 NADH formed 2 ATP, so 2 ATP. Okay, so and if we talk about the 1 NADH, so that make the 3 ATP. 3 ATP. So 1 NADH, 3 ATP. We can multiply this by 3. That will be 24. And 1 NADH is making 2 this one. So we can multiply 2 multiply by 2. And if we talk about the 1 FADH, that is basically the FADH that is making also the 2 ATP. So we can multiply 2 multiply by 2. The answer will be 4, 4, 4. So these are what? These are the 32 ATP. 32 ATP. 4 ATP will be in the next topic we will be discussing, that 4 ATP will be formed from the substrate phosphorylation. So that 4 will be adding in the 32, so the 36 ATP will be formed from that electron transport chain. Okay, so we are having the 30, as you guys all know, we have discussed that the in the electron transport chain there are 36 ATP formation. So 32 are there, and that will be synthesized by the you can say ATP synthase. In the next video we will discuss about the substrate level of the phosphorylation, in which we will be discussing that we will be having the 4 ATP will be formed there. So that will be at, you can say, if we will add 4 into 32 which is formed by ATP synthase, that will be the 36 ATP. In the next video we will discuss how the 4 ATP is basically formed.